Hello students, welcome to online classes. This is Swati madam, your biology teacher from Silver Oak School. Hope you are doing good. In the previous session, we have discussed about various activities to prove that certain raw materials are required during the process of photosynthesis and to prove that oxygen is produced during the process of photosynthesis. Later, we have discussed about TS of leaf, the diagram of TS of leaf, transverse section of leaf, and the exact location of the chloroplast, a cell organelle where chlorophyll pigment is present. Today, in this session, we are going to discuss about the structure of chloroplast. The structure of chloroplast is very important to know, and here we can know where the light dependent reactions and light independent reactions will take place. Let us see the diagram now. Now let us learn the diagram of chloroplast. First you have to draw oval shaped structure. It should be double line. Oval shaped structure. Inside that we have to draw thin long thread like structures like this. 3 or 4. Onto which we have to draw thylakoids and it should be connected with each other. So first draw thin thread like structures. Onto this we have to draw thylakoids like this, rectangular shaped cells. It should be piled up one another and it has to be connected with the long thread like structures. Here and there we have to draw the thylakoids and all the thylakoids together will pile up to form a structure known as granum. The major part where light dependent reactions takes place during the process of photosynthesis. After that, we have to draw the starch granules which are present here and there in the chloroplast, a circle and few circles inside. And here inside the empty portion of this double membrane structure, we have to mention loop of DNA. And also it has to be shaded. Either you can keep dots, some dots, or you can shade it, the structure. The intermediate fluid portion which is present inside the chloroplast, we call it as stroma. We have to shade that portion, or otherwise you can keep thin dots. So here is the structure of chloroplast. You can draw it in an easy method. Every time we have to draw the diagram inside a box and should be represented the parts separately for better presentation. This is the structure of chloroplast. The parts I have mentioned here. It is a double membraned organelle. You know very well from your 9th standard, chloroplast is a double membrane organelle. Now, it consists of outer membrane and inner membrane. The fluid space which is present inside the chloroplast is known as stroma. This is known as stroma. The matrix which is present inside the chloroplast is known as stroma. It consists of dots called as lipid globules. It consists of certain dots known as lipid globules, and here you can clearly see there are disc-shaped cells known as thylakoid. The single one is known as thylakoid. All the thylakoids will pile up on each other like this. It will pile up on each other in order to form granum. Here you can see the starch grains and loop of DNA also.
Chloroplast structure. At the end of this module, you will be able to describe the ultrastructure of chloroplasts. Chloroplasts are membrane-bound organelles that act as the main centers for photosynthesis. The number of chloroplasts varies widely in different plants. For example, in green algae like Chlamydomonas and Spirogyra, the number of chloroplasts present in each cell remains constant. In higher plants, the number of chloroplasts varies from cell to cell. Each cell may have 30 to 200 chloroplasts. Although the size of chloroplasts varies from plant to plant, chloroplasts are generally 4 to 5 micrometers in size. Also, plants growing in shade contain larger chloroplasts in their cells than those growing in intense light. Chloroplasts also differ in shape, being plate-shaped in chlorella, cup-shaped in chlamydomonas, ribbon-shaped and spirally coiled in spirogyra, and star-shaped in zygnema. In higher plants, most chloroplasts are nearly spherical or ovoidal in shape. Like most organelles in a eukaryotic cell, chloroplasts are surrounded by a bilayered membrane called chloroplast envelope and are disc-shaped. While the outer membrane is freely permeable to molecules, the inner membrane is selectively permeable and contains many transporters, such as the integral membrane proteins, which regulate the passage of small molecules like sugars in and out of the chloroplast. The inner membrane encloses a protein-rich ground substance called stroma. The fluid in the stroma contains a host of enzymes, plastid DNA, RNAs, and ribosomes. Within the stroma is another membrane system called the thylakoids. The three-membrane system divides chloroplasts into three distinct internal compartments. 1. The intermembrane space between the inner and outer membranes of the chloroplast envelope. 2. The stroma, which lies inside the envelope but outside the thylakoid membrane. And 3. The thylakoid lumen. The thylakoid membrane is a network of membrane-bound flattened discs called thylakoids, which are arranged in stacks called grana. The membranes of thylakoids are called fret membranes, and the grana are interconnected by tubular membranes called the intergranal lamellae. Each chloroplast may contain 10 to 30 granule clusters with a single granum comprising about 20 to 60 thylakoids. The thylakoid membranes are made up of 20 to 30% lipids and 70 to 80% proteins. They also contain a large number of granular structures embedded in the membranes. These granules are called quantasomes. Quantasomes are responsible for capturing photons of solar electromagnetic radiation and act as photosynthetic units. The larger granules are called photosystem II or PS2, and the smaller granules are known as photosystem I or PS1. The intergranal lamellae lack photosystem II but contain only photosystem I. The thylakoids contain photosynthetic pigments, namely the green pigments chlorophyll A and B, and the yellow to red pigments called carotenoids. Due to the three-membrane structure, the internal organization of the chloroplasts is more complex than in other organelles.
Now, let us discuss the structure of chloroplast. Chloroplast is a cell organelle, occurs mainly in the mesophyll cells of leaves. Most of the chloroplasts will be present in the palisade parenchyma and some few will be present in the spongy parenchyma. The size of the chloroplast is around 4 to 6 micrometers in diameter and 1 to 3 micrometers in thickness. It consists of three membranes. Actually, it consists of outer membrane and inner membrane, two membrane, double membrane, but it is believed that the third membrane forms a stacked sac-like structure. Here you can see very clearly, it forms the stacked sac-like structure one upon the another, and it forms a single one is called as thylakoid and together it is known as granum. Stacked sac-like structure, bag-like structure to form granum. The intermediate fluid portion, the shaded part which is present inside that is known as stroma, and this stroma will be similar to the cytoplasm present in the cells. And here in the stroma, generally in the granum, light dependent reactions will take place in the granum, which we are going to discuss in the mechanism of photosynthesis. Fluid portion which is present here, the shaded part, the inside fluid, we call it as stroma. And this stroma consists of Here enzymatic reactions will take place, and it consists of DNA, starch granules, and then all the synthesis of glucose will take place here in the stroma, and it leads to the starch formation. Substances which capture sunlight is called photosynthetic pigment. You know very clearly that chloroplast contains a pigment known as chlorophyll, which is aid in the process of photosynthesis. And this chlorophyll consists of an atom known as magnesium in the middle, which is similar to the structure of heme in hemoglobin. As you know clearly that hemoglobin consists of iron in that, and here chlorophyll consists of magnesium, which is similar to the structure of heme in the hemoglobin. And here the chlorophyll is classified as chlorophyll A and chlorophyll B. Chlorophyll A represents the blue-green compound, chlorophyll B yellow-green compounds. So children, it is very clear that chloroplast plays important role, and it consists of so many accessory pigments and chlorophyll pigment which plays a major role during the process of photosynthesis. So here it is very clear that there are so many events occurring during the process of photosynthesis. Here I have given the steps in photosynthesis. The first one is conversion of light energy into chemical energy. So here the light energy is converted into chemical energy. The second step is splitting of water molecule. Water molecule will be split into H plus and OH minus ions. Then the third one is reduction of carbon dioxide to carbohydrates. So here it is very clear that when the light energy has been captured by the plants, then so many reactions will take place even in the absence of sunlight. So first this light energy will be captured and it will be passed on to form so many reactions. Let us now discuss about the mechanism of photosynthesis. Just now we have studied events in photosynthesis. The first one is conversion of light energy into chemical energy. Second is splitting of water molecule. Third is reduction of carbon dioxide to carbohydrates. Now coming to the mechanism of photosynthesis, the stacked sac-like structure will pile up to form a structure known as granum in chloroplast. Here light dependent reactions will take place in granum. It is very clear that when the plants start capturing the sunlight, it will capture the sunlight, and after that this sunlight will be passed for the rest of the reaction. So there are certain events which will take place even in the absence of light. Those are known as dark reactions. These are known as dark reactions. So these are light dependent reactions, and these light dependent reactions will take place in a specialized structure called as granum in chloroplast. Light independent reactions means dark reactions will take place in the stroma of chloroplast. Please note down students, light dependent reaction takes place in granum, light independent reactions takes place in stroma. Light dependent reaction is also known as photochemical phase. It is also known as photochemical phase. Light independent reaction is also known as a biosynthetic phase. It is also known as biosynthetic phase. Note it down. Now, let us see the mechanism of photosynthesis. The first step here is light dependent reaction. These light dependent reactions are also known as photochemical phase. Light dependent reactions are also known as photochemical phase. These light dependent reactions will take place in the granum of chloroplast. Here in the light dependent reactions, there are three steps to be involved. The first step here is sunlight will be captured by this chlorophyll pigment, and it gets activated by absorbing photons. Photons is the smallest unit of light. This is the smallest unit of light. So the chlorophyll gets activated by absorbing photons in the first step. After getting activated this chlorophyll pigment, splitting of water molecule will take place in the second step. Splitting of water molecule means here the water molecule will split into hydrogen and hydroxyl ions. Note down children, splitting of water molecules will take place. Here it forms hydrogen and hydroxyl ions. Water molecules will take place, and this reaction is known as a photolysis. Very important one mark question. What is meant by photolysis? Photolysis. Here photo means light, lysis means breaking of the water molecule. Photolysis means breaking of water molecule, splitting of water molecule. Here the water molecule will split into hydrogen and hydroxyl ions in the second step of light dependent reaction. As this reaction is discovered by the scientist known as Hill, it is known as Hill's reaction. So here you can observe there are three questions. What is the second step in the light dependent reaction? That is splitting of water molecule. What is meant by photolysis? Photolysis means breaking of water molecule or splitting of water molecule. Here what are the ions formed? H plus and OH minus, means hydrogen and hydroxyl ions are formed in this second step. Who discovered this photolysis? Hill, and it is named as Hill's reaction. If they ask you what is meant by Hill's reaction, what is meant by photolysis, then you have to write splitting of water molecule. Now coming to the third step, the hydrogen ion and hydroxyl ions are formed in the second step. And this hydroxyl ion, OH minus, will convert into this water and oxygen, and water and oxygen will be released from the plant. As you know very clearly that water will be used up by the plant. This water will be used up by the plant and oxygen will be released into the atmosphere. Now coming to this H plus ions, these are involved in the dark reactions. The H plus and OH minus ions will be formed in the second step. The OH minus ions formed in the second step, here it will convert into water and oxygen. Water is used up by the plant. Oxygen will be released into the atmosphere. Now coming to this H plus ions, it is involved in the dark reaction. And here at the end of this light reaction, two important compounds known as ATP and NADPH. Note down students, very important abbreviations. ATP, adenosine triphosphate. ATP means adenosine triphosphate, very important. And NADPH. NADPH means nicotinamide adenosine dinucleotide hydrogen phosphate. I am repeating this. Please note down. ATP means adenosine triphosphate. Adenosine triphosphate. NADPH. Nicotinamide adenosine dinucleotide hydrogen phosphate. These are the two compounds formed at the end of light reaction, and it is also known as connecting link between the light reaction and dark reaction. Let me repeat what I have taught you now. Light dependent reactions are also known as photochemical phase reactions. These light dependent reactions will take place in the grana of chloroplast. Here in the light dependent reactions there are three steps. In the first step, chlorophyll gets activated by absorbing photons from the light energy. The source of light here is sunlight. Photons means the smallest unit of light. Here in the second step, splitting of water molecule will take place into hydrogen and hydroxyl ion. This reaction is known as photolysis. Photo means light, lysis means breaking. As this was discovered by Hill, it is also known as Hill's reaction. Coming to the third step, the H plus and OH minus ions which is formed in the second step is used up here in the third step. OH minus ions will be converted into water and oxygen. Water is used up by the plant. Oxygen is released into the atmosphere. This H plus ions, it doesn't do anything here, but it is involved in the next step, that is dark reactions. Here at the end of the light reactions, two important compounds are formed. ATP, adenosine triphosphate. NADPH means nicotinamide adenosine dinucleotide hydrogen phosphate. Now let us discuss about the second reaction, light independent reaction, which is also known as biosynthetic phase. As these are not dependent on light, it is also known as biosynthetic phase. It takes place in the stroma of chloroplast. Is it clear? Grana helpful in the light dependent reaction. Stroma is useful in the light independent reaction. Here in the light independent reaction, the H plus ions already formed in the light reaction. In the second step of light reaction will be used up by NADP, nicotinamide adenosine diphosphate, to form NADPH, nicotinamide adenosine dinucleotide hydrogen phosphate. You know very clearly that it has the full form I have given in the previous session. Now here in the dark phase, hydrogen of NADPH will be used up by the carbon dioxide and in the presence of ATP energy. Here what is the energy source? ATP energy source in order to form the glucose C6H12O6. Here this light independent reaction will take place with the help of so many sequence of events. Here so many intermediate compounds like RUBP and enzymes are involved. RUBP, the full form of RUBP, ribulose bisphosphate or ribulose 1,5-bisphosphate, because the presence of phosphorus groups at the first and fifth carbon atoms. Ribulose bisphosphate and enzymes are involved during the sequence events of this light independent reaction. Is it clear? So there are two steps here in the mechanism of photosynthesis, light dependent and light independent reactions. And here they have given clearly how the glucose is formed. Later it has been turned into starch in plants. These events will take place, the full events you can see in the glycolysis and Kelvin cycle, which you are going to study in higher standards. Now let us discuss about the parasitic nutrition in Cuscuta. As we learned before, heterotrophic nutrition has been classified into three types: saprophytic, parasitic and holozoic nutrition. Parasites, generally it will depend on the host plant. Either it may kill the host or it may not kill the host. Now let us discuss about parasitic nutrition in Cuscuta. Here Cuscuta, it is the genus name. Dodder plant which is given in our text book, it is a parasitic plant. It is a leafless, twining parasitic plant. It belongs to the family Convolvulaceae. And genus name here is Cuscuta. Around 170 twining species are present throughout the temperate and tropical regions of the world. How many twining species? Around 170. Here, it doesn't consist of any chlorophyll. So, it is very clear it doesn't consist of chlorophyll. So, it cannot prepare its own food material but depend on others for food material. But only in one species known as Cuscuta reflexa. Cuscuta reflexa is found to contain small amount of chlorophyll. Very important, remember, Cuscuta reflexa contains small amount of chlorophyll. So here the parasitic nutrition in Cuscuta, how it takes place means generally it after the seed germination, the seed will start germinating and it will produce the long slender stems. Later, it will start attacking the host plant. Whatever the plant which is present over there, it will start attacking the host plant. After it reaches the host plant, it will produce string-like structures known as haustoria. We call it as haustoria. Through that, it will absorb the water and food material. Haustoria. Through haustoria, it will absorb the water and food material from the host plant. It will just slender over the haustoria. It will first affect one host plant. After getting the nutrition, then it will later on continue its thin string-like structures all over the plant. Later it will continue to move to the other host plant. Here the leaves are reduced to spines, stems are slender and these are string-like structures. So many dodder flowers are present. These are cluster white bell-like petals are present in these dodder flowers. Generally, after attacking the host plant, this Cuscuta will take up water from the host xylem and nutrients from the phloem.
Let's start with parasitic plants. The plants which obtain their food directly from other plants are known as parasitic plants, and those plants from which they obtain food are known as host. You might have seen Cuscuta, amarbale, nearby you, which is an example of a parasitic plant. It coils around the stem and branches as a yellow tubular structure. You'll be surprised to know that it does not contain chlorophyll, so it takes ready-made food from the plant on which it is climbing. The plant on which it climbs is called a host.
Till now we have discussed about the plants, how plants will take up the raw materials and utilize it and convert it into products during the process of photosynthesis. Before we have discussed that there are two types of organisms, autotrophs and heterotrophs. Autotrophs are the organisms which can synthesize its own food materials. Heterotrophs are the organisms which depend on others for its food materials. Let us recall the thing once. Now, let us discuss about the mode of nutrition. According to the mode of nutrition, it has been classified into autotrophic nutrition and heterotrophic nutrition. As I said earlier, autotrophic nutrition is a type of nutrition where organisms can synthesize its own food material, for example, plants. Heterotrophic nutrition are the organisms which cannot synthesize their own food material, but it depends on the other organisms for its food material. Again, these heterotrophs will be classified into three different modes: saprophytic nutrition, parasitic nutrition, holozoic nutrition. Saprophytic nutrition. Saprophytic nutrition is the nutrition in which an organism obtains its food from dead and decaying organic matter. For example, fungi, bacteria. Parasitic nutrition. Parasitic nutrition is the nutrition in which an organism derives its food from other living organisms without killing it. For example, Cuscuta, leeches. The third one is holozoic nutrition. Holozoic nutrition is a nutrition in which an organism takes complex food materials into its body by the process of ingestion and then absorbed into the body. For example, human beings and other animals.
Fungus is getting its nutrition from the decaying and rotten matter. And this way of acquiring nutrition from dead, decaying matter is called as the saprotrophic mode of nutrition. Hence, it's not advisable to consume stale food or food after their expiry date, because indirectly it leads to consumption of harmful microorganisms, causing ill effects to your health. Let's move on to the next type of organism and see what exactly they feed on. We have all seen a parasite at some point of time. Now, parasites are organisms that live on the body surface or inside the body of another type of organism. For example, a leech is an organism that receives its nutrition by sucking the blood of another organism, while a roundworm lives inside the body of a human and receives nutrition from the human system by feeding on undigested food from our intestine. But please don't get worried. They are harmful only if they increase in number. Now, this way of acquiring nutrition from another living organism without actually killing the host is called as the parasitic mode of nutrition. Let's move ahead and see how some higher forms of animals acquire their nutrition. Let's group them according to their food preference. Lions, tigers, cheetah, fox are all flesh-eating animals, and hence we call them carnivores. While others like elephant, deer, rabbits, rhinoceros, cows and horses are all plant-eating animals, and plant or herb-eating animals are called as herbivores. And us humans, by our own individual choice, may consume plants or choose to have the cooked flesh of other animals. So, as humans can eat both plants and animals, they are said to be as omnivores. Now, all these higher forms of animals we just mentioned here have a proper digestive system. Hence, they are said to be using the holozoic mode of nutrition.
Here in the heterotrophic mode of nutrition, three different types we have observed: saprophytes, parasites and holozoic nutrition. Saprophytes, these are the organisms which will break down the food material outside and then absorbs it. For example, bread mold, yeast, mushrooms, etc. While coming to the parasites, it depends on other animals for its food materials, either by killing them or without killing them. For example, Cuscuta, lice, leeches, tapeworms. While coming to this holozoic nutrition, ourselves, it's an example. Depending on the food material, we take in the food and later we digest the food material. Is it or not? So, human beings is the best example for holozoic nutrition. Now here you have clearly observed the difference between this type of the food material and nature of the food material. Based on the availability, there are different types of organisms. While coming to this unicellular organisms like amoeba, paramecium, what about the nutrition there? There also the complex substances will be taken in and changed into the simpler substances. For example, let us see the nutrition in amoeba. Amoeba is a unicellular organism. It doesn't have a definite shape. You learnt about amoeba before. It doesn't have a definite shape. So what happens here? The food material will be ingested, means taken in into the body throughout the cytoplasm. It will be absorbed throughout it, and later here the food vacuoles will be formed, and then it will complex substances will be converted into simpler substances. Here the taking in of food will take place through a particular extension known as Pseudopodia. As you know very clearly, pseudopodia. It is nothing but the foot, which resembles a foot in amoeba. Here the pseudopodia will take in the food material and it will enter into the amoeba. It will be very useful. And now here I am going to show you an animation regarding the nutrition in amoeba. While coming to this paramecium, paramecium is also a unicellular organism, but it has a particular shape, slipper shaped. Generally we call it as slipper shaped. So here paramecium will take in the food material through a particular groove known as cytostome, oral groove. Through that it will take in the food material. It is covered completely with finger-like projections known as cilia. Now let us learn about the heterotrophic nutrition in human beings. Till now we have completed about plants. Now let us learn about the human beings. Here I am going to show you the nutrition in amoeba.
Nutrition in amoeba. The food and its way of obtain different from organism to organism. Example: amoeba takes food using fingers-like extensions of cell surface called as pseudopodia. They fuse over the food particle by forming a food vacuole. Inside amoeba, that food vacuole's complex substances are broken down into simple substances. Then, these simple substances diffuses into cytoplasm. The remaining undigested material is moved to the surface of cell and thrown outside.
Students, in today's class we have discussed about the diagram of chloroplast, structure of chloroplast, mechanism of photosynthesis, later about different types of nutritions, parasitic nutrition in cuscuta, very important topic, and then we have discussed about the amoeba, paramecium nutrition, unicellular organisms, and the different types of heterotrophic nutrition. So till here we have completed about the plant, photosynthesis, everything plant topic regarding the plants. In the next class we are going to discuss about heterotrophic nutrition in human beings. Hope you understood. So for better understanding, go through your textbook. All the best.